Lecture script with quizzes after every part. Part 1, genetic change, fast versus slow. In most animals, like mice or chickens, genetic change happens slowly. They need months to grow up, reproduce, and pass down mutations. That's a long cycle. But bacteria, they evolve fast. Why? Because they don't wait for the next generation. They can swap DNA directly with each other, gaining new traits almost instantly. So instead of waiting for offspring to evolve, bacteria can change within their own lifetime, using genetic exchange mechanisms like plasmids, viral transduction, and transposons. Mini review. Animals evolve slowly over generations. Bacteria evolve fast due to DNA exchange. Bacterial DNA can change during their lifetime. Quiz Part 1. Genetic change speed. Q1. Why does genetic change happen slowly in animals? Because they take time to mature and reproduce. Why do bacteria change genetically so quickly? A2. They can directly exchange DNA with each other. Q3. What are 3 ways bacteria exchange DNA? 3. Plasmids, viral transduction, and transposons. Part 2. Plasmids and DNA transfer. Plasmids are small circular pieces of DNA that float in the bacterial cell, separate from the main chromosome. What makes plasmids special? They're resilient. They survive even if the bacterium dies. They can be shared between bacteria. They often carry useful genes like antibiotic resistance. Think of plasmids as portable flash drives that bacteria use to copy and share powerful tools with each other. Mini review. Plasmids are small, extra DNA pieces. They can survive after a bacterium dies. They carry genes like resistance and can be shared. Quiz Part 2, Plasmids. Q1, what is a plasmid? A small circular DNA molecule separate from the main bacterial chromosome. Q2, why are plasmids resilient? They can survive even after the bacteria dies. What important genes do plasmids often carry? Genes for antibiotic resistance. Q4, how do plasmids spread genetics by being transferred from one bacterium to another? Part 3, F plasmids and the F pilus. Some plasmids are F plasmids, which have extra features. These allow the bacteria to actively send DNA to another cell using a structure called the F pilus. The F pilus is like a little tube or lasso. One bacterium builds the pilus, connects to a neighbor, and transfers the F plasmid. This process is called conjugation, bacteria having DNA contact. Mini review. F plasmids can actively move between bacteria. They use a structure called the F pilus. This transfer is called conjugation. Quiz, Part 3, F plasmids. Q1, what makes F plasmids different from regular plasmids? A1, they can transfer themselves through an F pilus. What is the F pilus? A tube-like structure used to transfer F plasmids between bacteria. What is the process of DNA transfer through an F pilus called? Conjugation. Part 4, viral transduction. Now let's talk about bacteriophages, viruses that infect bacteria. These viruses inject their own DNA into a bacterium. The host's machinery begins copying the viral DNA. There are 2 possible outcomes. 1, lytic cycle. The virus takes over, makes more viruses, and kills the host. 2, lysogenic cycle. The virus DNA hides inside the bacterial DNA and may stay dormant for years. Sometimes when the virus leaves, it takes some bacterial DNA with it. Then it brings that DNA into a new host. That's called transduction, and it's how viruses help bacteria share genes. Mini review. Bacteriophages are viruses that infect bacteria. Transduction happens when viruses move DNA between bacteria. 2 cycles, lytic kills and lysogenic hides and waits. Quiz Part 4, Viral Transduction. Q1, what is a bacteriophage? A virus that infects bacteria. Q2, what are the 2 viral cycles? Lytic, kills the cell, and lysogenic, hides in the DNA. What is transduction? Gene transfer from one bacterium to another via a virus. How can transduction lead to bacterial evolution? It allows viruses to transfer new genes, like toxins or resistance, to other bacteria. Part 5. Transposons, jumping genes. Transposons, or jumping genes, are DNA pieces that can move from one place to another. They can jump within one genome or between DNA molecules. This movement causes random mutations and increases genetic variation. Example, in Indian corn, transposons cause different color patterns in kernels. In bacteria, they can turn genes on or off, or even move resistance genes into plasmids. Mini review. Transposons jump between parts of DNA. They cause random mutations. They add to genetic diversity. Quiz, Part 5, Transposons. Q1, what is a transposon? A piece of DNA that can move from one part of the genome to another. What do transposons cause? A2, random mutations and increased genetic variation. Where are transposons found? In plants, bacteria, and all living organisms. Part 6, Recombination. In recombination, two pieces of DNA can swap parts if they get close and tangled, just like tangled headphone wires. Though rarer than plasmid transfer or transduction, recombination still happens and helps mix up DNA, creating new combinations of traits. Mini review. Recombination mixes DNA between molecules. It happens when two strands get tangled. Less common than other mechanisms, but still important. Quiz Part 6, Recombination. Q1, what is recombination? A1, the exchange of DNA segments between two strands of DNA. Q2, how is recombination like tangled headphone wires? The DNA strands get close and swap parts, like tangled cords getting mixed up. Q3, why is recombination important? It creates new genetic combinations and adds diversity. Part 7, why bacteria evolve so fast? Thanks to all these methods, plasmids, F plasmids, viruses, jumping genes, and recombination, bacteria can constantly change. Combine that with the fact that they reproduce rapidly, and you have a recipe for unstoppable evolution. One small DNA change can spread through a population within hours, especially in places like hospitals. That's why understanding bacterial genetics is critical for medicine, antibiotics, and public health. Final mini review. Bacteria use many methods to swap DNA. They evolve faster than humans due to rapid reproduction. These changes affect antibiotic resistance and disease. Final quiz. Recap. What 5 major mechanisms let bacteria exchange DNA? Plasmids, F plasmids, viral transduction, transposons, recombination. Q2. Why does bacterial evolution happen so quickly? Fast reproduction and constant DNA exchange. 3. What is the danger of bacterial genetic change in healthcare? A3. Antibiotic resistance can spread rapidly.